Good evening, everyone. I'm going to give it another few minutes just for the people that might be in lecture. I'm going to give them a little bit of time to try to join in, so I'll probably start in about 2 minutes just to let everyone know. All right, so I think I've given some people enough time. I'm sure other people will trickle in if they had just finished the lecture. Before we start, does anyone have any burning questions? I'm going to take that as a no. So then I will start. Today we are doing lectures 6 and 7. So lecture 6 should be a little bit of a review just because we had questions on it in your Friday Q&A quiz, and then I will go through lecture 7. If you noticed, I put up a picture of one of my previous study guides for lecture 7. It's kind of a study guide. It was one of my first attempts at condensing all the information onto one page. And it really only condenses the vaccine lecture, but we'll go through that. And then I have questions at the end on Poll Everywhere. I think it's maybe like 6 questions. Also, it is pretty important for you all to know for next week, on the Wednesday before your anatomy test, I will not be having a review session. I know Ivy sent out an announcement about it, but I just wanted to give you all the information. I'm not going to have a review session next week because I didn't want to add anything else onto your plate before your very first anatomy test. But after your genetics test, so the following Wednesday, I will have a review that night. And the next day, Thursday morning, I'm going to have reviews there that will cover lectures 8, 9, and 10. So your last 3 lectures. And then on Thursday after your last lecture for immuno before your test on Friday, that Thursday night I will have an open Q&A. So I'm going to do 2 reviews. And they're going to be the exact same. The Wednesday night after your genetics test, and then the Thursday morning after that, I will have a review. They're going to be the exact same information covering the lectures. And then Thursday evening after your immuno lecture, I will have a open Q&A for any loose ends or anything that anyone wants to cover. So just to make sure that you know, next week my reviews will be replaced with Justin's final review before the anatomy test. Also, it's important to note before I start, make sure you're doing your review sheets or your study strategies now. because I know once this weekend ends, everyone's going to be focused in pretty much only on anatomy. So try to get as much studying and prep work done now that you can for immuno, because you will not have time if you leave it until the end. And if you try to leave it until the end, it will be reflected in your test grade. That happened to a lot of people, an overwhelming majority actually, for the first immuno test. All right, so lecture 6, immune responses to infectious agents. It's pretty straightforward. Like I said, this is when we start getting to the more interesting things, in my opinion. The learning objectives, make sure you go through them. Like always, make sure you use them to establish your review sheets or your study sheets. The learning objectives for the next lecture are a little bit wonky, and we'll go through that. She didn't directly cover one topic in the learning objectives for lecture 7, so we'll go through that when we get there. But in this one, these are all very straightforward, and make sure you know and understand all of these. All right. So immune response defense against microbes. So we kind of already know this. We have our innate immunity, which is just going to be our natural barriers. So our PAMPs and then our PRRs, our basic innate phagocytic cells, so PMNs, neutrophils, and macrophage. And we have the NK, and then our complement system. Remember, for innate immunity, our complement system is going to be the the alternative complement system. So for innate, it is the alternative complement system. Then our adaptive immunity, just like always, our T cells, which is our cellular response, our B cells, which make our antibodies, which is our humoral, and then here we have the classical complement system. So make sure you know which complement system goes under which type of immunity. All right, here we're just talking about how diseases can be caused by microbes, but diseases can also be caused by the host response to these microbes or pathogens. And we've talked about this before, how inflammation can cause the bystander effect. Chronic inflammation can have deteriorating effects to nearby tissues. And then in the last lecture, we talked about the 4 different types of hypersensitivity. The effects of the antibodies. So this is pretty important here. We have the first main effect of the antibodies is the initiation of the classical complement system. So that's going to be in our adaptive immunity. Then ADCC, which is antibody-dependent cytotoxicity. And we're going to talk about this a lot. And this one is pretty important because this is how we use antibodies in any sort of viral infection. This is also how we use antibodies in immunotherapy for cancer treatment. And we're going to get into that. Then neutralization. So neutralization, antibodies essentially binding to either toxins or microbes. In either case, neutralization is just stopping the action of. So antibodies will be stopping the action of either the microbe or the toxins. And then opsonization is just flagging microbes for phagocytosis. So it's antibodies attaching onto microbes and flagging them and marking them for phagocytosis. So we have 2 ways of killing here with antibodies. Well, 3 ways. We have the complement system, and then we have ADCC, which is antibodies flagging them for cell death. And then we have opsonization, which is antibodies flagging microbes for phagocytosis, which in turn leads to cell death. All right, so this is going to be another little bit of a review here. We've seen this already with the complement system, that IgM or IgG binds to the microbes, and it's all going to be initiated by C3. C3 will start that zymogen cascade, that cascade of cleaving proteins all the way until we get to the C5 to 9 complex. So C3A is going to lead to 1, C3B. Here you can note C3B is for an amplification loop. So what that means is that's actually going to amplify here the alternative pathway. So make sure you note that we've talked about C3A before, and we will talk a little bit about C3B a little later on. So our classical complement system here, once again, its entire purpose is opsonization, neutralization, inflammation, and lysis. This is what it actually does. Lysis is one we've already seen. I've already explained how it creates that pore, that hole mechanism in the membrane, which will then cause the infected cell or microbe to go through apoptosis. And then inflammation, neutralization. Inflammation, the complement system being used as a sort of a cytokine system, where the complement system is going to initiate recruitment of other innate immune cells, so macrophage and neutrophils. And then neutralization, opsonization is something that makes kind of sense that we're going to get into. We've kind of explained already. So ADCC here. ADCC is when we are using antibodies to bind to target cells, whether it be a microbe or a virally infected cell or a tumor cell. And the antibodies bind using their FAB region. So it uses the variable chain and it binds to our actual target. When it binds, it leaves its FC region exposed. So the actual constant region, the heavy chain constant region is exposed. What happens is once we get that binding of the antibody and we have that constant region exposed, then an effector cell, in most cases, we're going to be talking about NK cells being the effector cells for ADCC, but almost any cell can do it. So an NK cell, a monocyte, macrophage, neutrophil, eosinophil, dendritic cell, a T cell, a lot of these cells can take part in ADCC, but we're going to mainly be talking about NK cells. So the NK cell or any of these effector cells have an FCR. So that's a receptor for the FC region. So that FCR binds to the FC on the antibody that's exposed, and then that triggers the release of these cytotoxic granules. So we've already talked about these. I've already given you examples of these. These are the porphyrins and the granzymes. These porphyrins and granzymes, these cytotoxic granules, will interact with the infected cell or the target, and then it will force that target to actually express cell death inducing molecules. And these are molecules that just induce apoptosis, and they induce mitochondrial apoptosis. So it's apoptosis through ROS, through reactive oxygen species. It is important to note that ADCC is non-phagocytic. So our phagocytic line with antibodies is going to be opsonization. But our non-phagocytic, so our direct killing line, direct killing always results in cell death, is going to be ADCC. We've got a beautiful image here of what's occurring. So we have here on the left a virally infected cell, and we have these little IgG antibodies. These IgG antibodies, the FAB region, so as you can see, that's the light chain, so the tip of the Y portion, actually attaches on to these little epitopes. So epitopes are just anything that's on the cell surface. So it attaches on to anything on the cell surface of this virally infected cell. Then we have that FC, that constant region, exposed. That constant region will then attach onto the FC receptor, and then we'll have a signal propagation that will release these porphyrins and these granzymes. These little bars here, these are just normal modulators, they're normal cytokines that will actually go through and create that hole in the membrane. By creating that hole in the membrane, very similar to the complement system, it forces this cell, this virally infected cell, to undergo and force itself into apoptosis so that it dies. So remember, ADCC always ends in cell death. That is the entire purpose of it.
Hey, Daniel, for the antibodies, are these just free-floating antibodies that are working with this?
So they can be free-floating. They also are usually made. So these are antibodies that have high affinity maturation, which means we've already gone through isotype switching. So we've gone through converting it from IgM to IgG. And then we've also gone through affinity maturation, making it specifically for this. So most of the time when you see this, this is going to be something that we have for like vaccinations. So if we have a vaccine, and we're going to get through it, but you've kind of already seen the information with the lecture with Dr. Anderson. A lot of the vaccines for viruses only produce antibodies through the mechanism that they actually use to create that established immunity. So those antibodies that we create through those vaccinations are these antibodies here in ADCC.
All right. Just to kind of follow up on that, because let's say the B cells are stimulated by the helper T cells, right? Mm-hmm. To make the antibodies. Wouldn't the T cells have to be stimulated by some like dendritic or macrophage cell to? Yeah. So then there is but then those cells are going to phagocytize the antigen, right?
Yes. Mm-hmm. But that would be on an MHC class 2. So right. This one on a virally infected cell for here, this would be going through MHC class 1, and that would directly target a CD8. This is more when we have memory T cells and memory B cells activating.
Got it. Okay.
So this wouldn't be on that initial encounter with antigen, which is the MHC class 1, class 2. That's all initial encounter. Here, ADCC is once we already have antibodies made from memory B cells, then they're going to establish this ADCC. So this is how you have that long-term immunity.
That makes sense. All right. Thank you so much.
No problem. All right. And I assume that answered the question, Suhas. I hope I didn't butcher your name. Suhas's question in the chat. So the antibodies being used, the main antibody being used here is going to be IgG. That's going to be our main workhorse, and that's going to be the main one that induces cell death throughout this process. And we'll get into that a little bit more. And I believe she mentioned that as well. But these are antibodies that we've already made and used for long-term immunity. All right. And then, so NK cells with ADCC, once again, this is going to be used for intracellular bacteria, intracellular viruses, and then tumor cells. So any cell that's experiencing stress will actually express certain epitopes on its outside that antibodies can interact with, as well as anything that has an intracellular bacteria or intracellular virus that we've already been exposed to. They will kill the cells directly. It can also secrete, the NK cells can also secrete interferon gamma. That's what this little IFN and then gamma symbol here. And interferon gamma is used similarly to a cytokine to actually attract macrophage. And that's going to induce an inflammatory response. So that's pretty important to note here that that's another option of what NK cells can do. So as we have this process and the antibodies bind these epitopes and then they recruit that first NK cell, which is going to stimulate cell death, the NK cell will also recruit more macrophage to the area. And it does this so that way it can have reinforcements. These are not directed reinforcements. So it's not actually calling snipers that are going to say, hey, shoot this exact target. It's just calling someone with a huge heavy machinery and just say, hey, we have one problem here. I don't care who you get rid of, but you need to get rid of a lot of things here so that way we don't have any sort of infection. It is important to note that these granules do induce apoptosis. And we talked about that with the porphyrins and the granzymes inducing apoptosis. You don't need to know the exact name of this receptor. She does go through the process of saying that this FCR is also known as FC gamma R3 or CD16. She's most likely on a test not just going to put CD16. She's most likely going to put FCR and CD16 or FCR and FC gamma R3. So just be familiar that the FCRs on NK cells have multiple names. Make sure that that when if you do see CD16 on a test question, you don't get thrown for a loop and you kind of have an understanding of what she's referring to in there. And then we've talked about the release of the granules, and she's going to have another picture about it, which is the same process that we've talked about. She does, as you guys have noticed, she does like to use a lot of imagery, so that way she can hit all the different ways that people kind of either view things or understand things. So this is that same image. So you can see here we have IgG attaching on its variable region. So the FAB region is attaching onto certain epitopes, these little surface antigens, onto this infected cell. And the FC region will interact with the FCR on the NK cell, and then that will trigger that release of the porphyrins and granzymes to then induce apoptosis of the actual infected cell. Yes, Cameron, so CD16 is another name for FCR. And we're going to get into some other names, some other CD names for other receptors that we haven't really talked about, that she just kind of throws them at you all and hopes that you all know what it is. So just make sure that you know that they have multiple names and these names are interchangeable. And then another example is IgE. Remember, IgE is used for helminth infections, so worm infections. It's also used for any sort of allergy. So it's going to go through the same process. IgE can bind with its AB region, and then its FC region will bind to the receptor and on the effector cell, and it'll go through a specific response. So if it's IgE and some sort of allergen, so if the IgE is binding an allergen here instead of a pathogen, then that a mast cell will attach, the FC region of a mast cell will attach onto the IgE and will cause an increase in histamine release to give you worse allergies. So this can be done this ADCC can be done in multiple different ways. Our second form of activity for antibodies, neutralization. So like I said, neutralization, the entire purpose of it is just to prevent something from occurring. So whether we're blocking the action of toxins or whether we're blocking the infection by a microbe. So what I mean by blocking the infection by a microbe, this is to stop the microbe from entering and infecting cells. So that's kind of what we're doing here. And she has multiple pictures to go through it. And the same thing with blocking the toxin. So if we have an antibody and it's binding to a toxin, that will prevent that toxin from entering the cell and actually going throughout its biological activity. So inducing whatever it's going to, whether it be affecting cell signaling or protein synthesis. And this is something that we're going to see a lot of in the 7th lecture for vaccination. So the process that it actually goes through, the microbe enters the host, and molecules and envelopes or cell walls bind to the host cells. Antibodies will attach to the surface molecules of that microbe, and it will prevent entry of that microbe. Same thing happens with the toxins. So the toxin will attempt to attach onto glycoproteins on our cells' surfaces. If we have antibodies for that toxin, so if you've received certain vaccinations that will create antibodies for toxins, and we'll go through that in the next lecture, those antibodies will be free roaming because you already have them. And those antibodies will then bind onto those toxins and prevent the toxins from binding onto our glycoproteins and stop them from eliciting their response. And we've got a beautiful image here for the toxins. So we can see this little yellow shape with a red circle on it. That's going to be our toxin. So the toxin will bind onto a glycoprotein receptor, and then it'll go through endocytosis, get inside, and then release its effector, which then poisons the cell and whatever it's going to do. Like I said, whether it's inhibiting protein synthesis or stopping cell cycling or increasing C-amp, whatever these toxins might do, they're all very different, and you'll get into them a lot more in micro and virology in the next few blocks. But if we have that antibody that's specific for that toxin, that antibody will go around and it will bind the toxin, and it will prevent the toxin from actually attaching onto those glycoproteins to enter. It does the exact same thing with the microbe, and I believe she has a picture of it on the next slide. Action of the... So ADCC is a form of activity of antibodies. So it's a specific type of action of antibodies. So not necessarily the complement system, but ADCC is antibody-driven cellular cytotoxicity. So it's when we're using antibodies to specifically kill microbes or pathogens. To answer Marie's question in the chat. So here she has a whole bunch of, once again, a lot of pictorial demonstrations here of what our antibodies can do to neutralize. So the first three are all revolving around preventing microbes from entering or getting past certain barriers in the body, and the last one is going to be the toxin. So the first one, we have a microbe trying to get past an epithelial barrier. So if we have a whole bunch of antibodies to that, to specific epitopes or glycoproteins or anything on the surface of these microbes, the antibodies will attach, build a complex, and we've seen this before, this antibody antigen complex. They'll build a complex that's too large to get through the epithelial layer. Same thing is going to happen if it's trying to enter into a cell. If it's trying to enter into a cell, if we have one microbe trying to enter in a cell, we'll build that antibody antigen complex, and it won't be able to attach onto the cell's receptors. If we have a cell that's already infected, and normally when it's infected, the cell will then lyse, so it's going to break open and release all the new microbes or pathogens. So then those antibodies will attach on and prevent those newly created microbes or pathogens from attaching onto new cells. So still, same process. Neutralization is just to stop it from doing its action. It's just to prevent the microbe or pathogen from doing whatever it's trying to do. And the exact same thing with the toxin. And they're binding in the same place. The FAB region is still what binds to the foreign body. It's still what binds to the toxin. It binds to the microbe. So the FAB region is what's going to bind and prevent the action here. All right, so then opsonization. Opsonization is just phagocytosis. Remember, when you hear phagocytosis, and this was a slight confusion that came up in the early morning review, and I understand why, because it sounds confusing. Phagocytosis doesn't automatically mean death. What phagocytosis means is just eating. So it's a macrophage or a neutrophil or a dendritic cell just eating a target antigen. That is it. phagocytosis should lead to that microbe or that antigen or that pathogen being put into the phagolysosome for destruction. But it doesn't always lead to that, and we'll get into that into a little bit. But when you think of opsonization, just think of phagocytosis as in the process of eating the microbe. So opsonization for antibodies is just targeting this microbe for phagocytosis. So targeting it for it being taken up by a macrophage or a neutrophil. So we'll have these extracellular bacteria, and they usually, in this scenario, are going to be encapsulated. The reason it's important for encapsulated bacteria is because when you are encapsulated, when you have that capsule around you, you have a lot of different epitopes on your outside, a lot of different virulent factors that these antibodies can bind to. So it gives us a higher chance to be able to create antibodies that can bind to those epitopes because there are more of them. So we'll have this extracellular bacteria, and we've created these antibodies, usually IgG. Remember, IgG is the workhorse of the antibodies, so of the immune globulins. It is the one that does the majority of the work. So IgG being made already through B cell and T cell interaction with class switching. We create a whole bunch of them, and they bind to the outside of this microbe, just completely covering it, making that complex. That complex then is forming a target for this macrophage. So we'll have interaction between the FC region of the antibody and the FCR on the macrophage. That will then lead us to that phagocytosis. So it's going to lead us to engulfing that microbe. Once we do, then we can send it to that phagolysosome and move it off for digestion. So here's some immune cells, what they do to microbes. This shouldn't be anything new here. Phagocytize, recruiting other cells. We've talked about all of these little things, using granules, presenting antigens. We've talked about this. This has been the past 5 lectures. And then the immune response can increase pathogenesis. We know this as well. We've talked about this a little bit. We've talked about COVID and some other things leading to that cytokine storm and that bystander effect. So remember that bystander effect is just when nearby tissues get affected by our immune response that we're actually trying to do. In this case, for bystander effect, that immune response is pretty widespread. It's not targeted, and it's having that effect on these nearby tissues and destroying them and stopping the function of nearby tissues. Some of the ways this can happen is through an overproduction of cytokines. And we kind of mentioned this a little bit when we talked about COVID a few last review or before that, I'm not sure. But we talked about it with the cytokine storm. And that's really the biggest problem that we're seeing with COVID is that actually we have such an increase in cytokines being produced by our immune cells that these cytokines are actually causing an altered mental status, fever, tachycardia. They're causing a lot of respiratory issues. So we're getting increased inflammation in the bronchioles, which is causing a lot of problems. That's why people are having to go on respirators. We can also see it in other forms with leading to bacterial septic shock and food poisoning. You'll go into 2 really strong examples of that in block 2, so in a few weeks. And then also we're going to talk about superantigens. That's something that that she kind of mentioned and glossed over. It's going to be something that you hit more so again in block 2 with Dr. Anderson. So the superantigen, the one that we're specifically going to talk about and the most common being staphylococcal superantigens. So what it actually does is when you have presentation between the MHC class 2 and the helper T cell for this bacterial infection, you have your normal antigen. But then it will also, these certain strains, mainly Staph aureus, will create a superantigen. And that superantigen actually attaches on to the MHC class 2 and the TCR. And it kind of holds it there. And what it's doing is it's forcing the T cell to create a response and to release cytokines, but it's not releasing a targeted response because we're not getting proper conversation and proper presentation between the MHC class 2 and the TCR. So we'll get a better understanding of this in the picture. Just note that we are presenting an antigen normally, and then we have the superantigen as well attached onto our complex. But what we're not getting is that antigen crossing over into our T cell for the T cell to then present it to a B cell. We're just getting a huge increase of cytokines. And we'll see that a little bit more in this picture. So in this image, we can see we have normal antigen from these staph, and it's being presented from an MHC class 2, which it would because it's exogenous antigen created by an extracellular bacteria. It's being presented to our TCR on our CD4 cell. Normally, this antigen would go from the MHC class 2 to the TCR, as long as we have that co-stimulatory response. It'll go into the CD4 cell. The CD4 cell will then process it and present it to a B cell. But that's not happening because we have this superantigen. The superantigen is attaching onto the actual receptors separately. So not in the cleft of the receptor, but it's attaching on the outside of the receptor. What that's doing is that's holding this still and forcing this T cell to release more and more cytokines without actually interpreting the normal antigen signal that it's supposed to be getting. So with that, we get that huge cytokine storm. So some other things that we might need to note: bacteria that hide within cells. So these can be phagocytized, but they don't go through that process of being taken to the phagolysosome. So make sure you note that. These are usually viewed as intracellular bacteria. There are other examples of intracellular bacteria you're going to get into with Dr. Anderson, but make sure you note these. And think about it, if they are intracellular bacteria, if they enter the cell, but they don't get destroyed, and then they start to produce their byproducts, whether it be toxins or whether it be going through replication, they're producing those within the cell, which now forces these to actually produce an endogenous antigen, which will lead into an MHC class 1 pathway, which is different than what we normally think because they are bacteria. But remember, the antigen is always specific. It's not specific to whether it's bacteria or virus. It's specific to where it's being made. So if the antigen is being made within the cell, that's going to be an endogenous antigen, leading to MHC class 1 and activation of CD8 cells. If the antigen is being made outside of the cell, then it's going to be an exogenous, leading to MHC class 2, leading to CD4 activation. So make sure to note that and know those 5 different genuses. So we're going to talk about some virulence factors of some of these bacteria that help them to not only avoid our immune response, but also kind of control our immune response. And it's an evolutionary advantage that they have, that they've gone through over time, that they've done simply just to survive longer and create their virulence. So the first example, and you're going to talk about all of these in Dr. Anderson's in his block, in the next block, but the M protein. So M protein is found on the surface of a Streptococcus pyogenes. So there's going to be a gram-positive here, so it has a very, very large capsid. And just below that capsid, we have the M protein. So it is on the cell surface, but it is below our capsid. So the M protein, what it actually does is it prevents phagocytosis. It also prevents binding of the C3b portion. Remember, C3b is the secondary portion in our complement system. So this can avoid the complement system and our normal innate cell activation, which is just phagocytosis. So make sure that you note that. B cells can bind to it and make IgM, but they can only make IgM because there's no formal presentation, because it avoids that phagocytosis from the APCs. Our next one, protein A. Protein A is found in Staph aureus. And what it does on the outside, it prevents binding of IgG. So that's pretty important because that's going to stop opsonization. That's also going to stop neutralization. And that's, for the most part, going to stop ADCC. Because if we can't bind IgG to it, then we can't activate our ADCC pathway. And this is one of the reasons why Staph aureus is such a big player in infections. When you hear of things like MRSA, you'll see a lot of staph strains that are actually resistant to antibiotics, and some of these things attribute to that resistance. All right, so our summary, remember, use her summaries specifically to build your study sheets and your notes. It's very important you guys start working on those now. If it was me, I would make sure by this weekend, by Sunday, that I would have every single note sheet done up to lecture 8. Which is going to be the lecture that you're going to have tomorrow. So that would be my suggestion, to have all those done. So once you finish anatomy test, you only have 2 more note sheets to make for immuno, and you can concentrate a lot of your focus onto biochem and genetics. That's just my suggestion. So antiviral immunity. So viruses are small infectious agents. We call them agents because they are non-living. They're non-living because they need the host to replicate. So they actually need the host cell to replicate, and they replicate inside living cells. So they have the innate immunity and the adaptive immunity for viruses. So our innate immunity is going to be interferons, which is just going to be producing cytokines, and then NK cells, which we already know we talked about with ADCC. Adaptive immunity, just once again, is our antibodies that can either prevent infection through neutralization, or it can go through the process of ADCC. And then we have CD8, which is our cell-mediated immunity. So here's just some examples. We've seen this a lot already. We've seen the adaptive immunity go through with neutralization, as well as CD8 cells targeting. And then we've seen it with NK cells. NK cells here, this is not going to be ADCC because we don't have antibodies working in this image, but this is just a normal NK cell finding a stressed cell, which would be an infected cell, a virally infected cell, and targeting it for apoptosis. And then we can see here also interferon gamma working on that antiviral cell to prevent viral infection. So evasion of the immune system by viruses, we have some specific examples that I would make sure to note. Viruses do a very good job of downregulating the MHC class 1, and they do that in different ways. Our largest example is going to be herpes. So herpes, it evades the immune system in two ways. It can inhibit inflammation by blocking the production of cytokines. And this is how herpes stays latent in your cells. So this is why you can never get rid of herpes. Once you have a herpes infection, you're always going to have it. It's not going to always show because the virus isn't always in its lytic stage. It's not always in the stage of replication, but it does a great job hiding within our own cells. And it does that because once it infects the cell, it can prevent the cell from releasing cytokines. And if we don't release the cytokines, then we're not getting that inflammatory response. We're not getting the recruiting of other innate immune cells. The other thing it can also do is it can interfere with the TAP and Tepacin interaction. So remember that TAP Tepacin interaction was crucial for expressing MHC class 1 to target and initiate that CD8 activation. So by interfering with TAP, we no longer get that presentation from MHC class 1. Remember, MHC class 1 is available on every nucleated cell. So it's important to note that herpes has that ability to actually downregulate that MHC class 1 and evade the immune response and actually suppress the immune response. The other example that we're going to talk about, it's a little different, but it's HIV. So HIV causes immunosuppression. And the way it actually does that is the target for infection for HIV is CD4 cells. So because viruses all go through the same lytic process, which means they enter the cell, they replicate, they take over the host machinery, they replicate, and then once they've replicated enough to a point, they actually force the cell to explode. Because they go through that process, when HIV is doing that, it enters into a CD4 cell, and it'll go through the process of replicating until it lyses that CD4 cell and causes that CD4 cell to die. So not only are you having the problem of this infection, but you're also having the problem of you're losing your helper T cells. So you can't actually build an immune response to extracellular bacteria. And that's something we're going to get into at the end of this lecture here. So just some images on the process with TAP Tepacin here. So if we're inhibiting this TAP here, we don't get presentation on the MHC class 1 in the endoplasmic reticulum and then transported all the way to the cell surface. So make sure you note that, and that's pretty important with that herpes virus. And then another image of the same thing, just slightly different with a little less stuff on it. Once again, if we interfere with that TAP, we lose that class 1 presentation. So here's some viruses that actually can lead to pathology through our immune response. So immune cells being affected through HIV, we just talked about that, and that also allows the HIV to spread more effectively by infecting immune cells. So if it infects the immune cells, we know we have immune cells that can travel throughout our entire body to build an immune response. This allows the HIV to travel throughout the entire body and replicate and find new cells that it wouldn't normally be able to target unless it's infecting an immune cell. Then we have some other ones, rhinovirus and influenza. These just cause that increase in cytokines, which gets you those flu-like feelings. So the achiness, the fever, the chills, these are all things that are caused by the cytokines. So we have an increase in cytokines from these actual viruses that gives us that pathology, that feeling of being sick and weak, and that feeling of malaise. Then we have Epstein-Barr and Coxsackie virus. You're going to talk a lot about Epstein-Barr creating an autoimmune disease. It's going to be something that's going to be pretty important in also block 2 with Dr. Tang. So our summary to viruses, now we're going to get into fungus, and then we'll get into protozoa. So fungus, our antifungal immunity. Fungi are eukaryotic organisms, so they're very similar to us. They have very similar machinery. There are some things that vary between us and fungi, and these are the things that we can kind of target either through treatment or through our own immune system, and we'll get into those. So the innate targeting for fungi, so the way our innate system deals with it, is neutrophils and macrophage. And this is going to be pretty much the largest way that we deal with fungi, mainly neutrophils. But both neutrophils and macrophage, and the way they can actually kill any sort of fungi is through ROS. So remember, that is their one way of actually inducing cytotoxicity, is through the reactive oxygen species. And then our adaptive immunity, it's mainly going to be CD4. CD8 can do a little bit, but not that much. Now, being that it is CD4, so it's helper T cells, if we have a higher Th1 response, that's going to actually destroy the fungi more. So remember, Th1 is going to be an increase in cell death, and then Th2 is actually going to be an increase in an inflammatory response, an allergy response. So with fungi, and most fungi infections actually end up being respiratory. With those fungal infections, with that high Th2, we'll have an increased allergy response or an inflammatory response, which, as you should be able to tell, can cause some serious problems when we're looking at respiratory infections. So like I said, fungi are very challenging to treat, mainly because they are so similar to us. There are some differences that are important to note, and we'll get to those on the next slide. And it's also important that they are aerobes or facultative anaerobes, so they can live in both environments. So you can have fungi that can live in the respiratory system, or you can have fungi that can live in the gut, which has no oxygen. And it's also important to know, most of you should already know this, they're saprophytic. What that means is they break down, they decay dead organisms. So that's a pretty common one. That's common knowledge, but this might be a term that many of you might not have seen. So these are the special parts about fungi. These are the things that we're going to target mainly with therapeutics and our immune system. So in the cell wall, we have chitin, mannan, and glucan. And in the cell membrane, we have ergosterol. Ergosterol is going to become a huge player when we get into block 3 of immuno, when you talk about fungi and parasitic immunity and how we actually deal with them in treatments. You're going to see ergosterol is actually one of the big players in our treatment form. Chitin, mannan, and glucan we use mainly for our own immune system to regulate and kind of find the fungus, but the ergosterol is used mainly for treatment. Mannan is going to be a PAMP that we see normally, and we kind of mentioned that before with the mannose pathway when we talked about PAMPs and PRRs. I know she kind of glossed over this and said it's not that big of a deal, and it really, this should be one of those things that you kind of study a little bit and you leave on the back burner. It kind of makes sense. So a superficial fungal infection is going to be on the most superficial layer of your body. Cutaneous and subcutaneous, we're going to see this in the dermis, whether it be in the epidermis or the actual dermis, so a slightly deeper skin layer. And then systemic or deep, that's going to be something internal organs. So this isn't that challenging of a slide here. It's just information you should be able to process just by seeing this prefix, whether it be superficial, cutaneous, or what have you. So once again, for superficial specifically, for the innate immunity, we have these PRRs and these PAMPs. The PAMPs being the mannan, so that's going to be the mannose that we see, and that's what allows our macrophages and our neutrophils to interact and attack them. And then we have our adaptive immunity, so our antibodies and cell-mediated response, mainly going to be antibodies, in this case for fungi. And then there's 2 specific examples that she gives, and I would make sure to note these. These historically have been pretty important come test time, from what I remember, even though the test changes every year. We have 2 examples. We have AIDS patients and cancer patients. Both of these should make sense because in both of these cases, we have a decreased immunity. We have immunosuppression. So with an AIDS patients, because we have decreased CD4 cells, we actually have a likelihood of getting pneumonia from this pneumocystis fungi. And then with cancer patients, because they go through chemotherapy, they're what's called neutropenic. So they have a very low neutrophil count. And neutrophils are used very much in our fight against fungi. So cancer patients are actually at a very, very high risk for an aspergillus infection, especially in Florida, because aspergillus is a very, very common mold and fungus that you see in Florida. And Dr. Safang will go over that in block 3. But make sure you have these two examples in the back of your mind and you remember them, especially come test time. So some of our other examples, protozoans and worms, we've kind of touched on worms a little bit. So protozoa, these are unicellular eukaryotes, and then worms are multicellular eukaryotes. We have a very weak innate immunity response to them. We actually need antibodies for this process, and we'll talk about it. You should already know and kind of remember that we use IgE for worms, also known as helminths. And we'll get into that in an image, I think, in the next slide after this. So some examples to remember on how these protozoas can evade the immune system. So Plasmodium, specific species of Plasmodium have antigenic variation. What that means is the epitopes on the outside, any sort of receptor that they have on the outside, they can change the genetics of those receptors. So they can change the molecular composition of those. And that makes it very hard for our body to create antibodies. So if we create an antibody with a very high affinity maturation, something that's going to really target that epitope, that receptor that they have on the outside, and then it goes through a generation and it changes what that epitope looks like, that antibody we made is just completely useless. So that's something that's going to be pretty important to remember. And that can also cause molecular mimicry because we're trying to create so many antibodies to keep up with how they're changing the epitopes on their outside, their receptors on their outside, that we might create some that actually recognize some of our own receptors. And this is something that we see a lot with Plasmodium. And then Trypanosoma cruzi, both of these you're going to hear again in block 3 with Dr. Safang. Trypanosoma cruzi, it has a innate ability to block IgM antibodies. So it can stop IgM antibodies, and it can also it can bind to macrophage and prevent macrophage activity. So these are important examples to remember. You don't need to know them in too much detail, but once again, have them in the back of your mind. So with protozoan and worms, so protozoan here at the top, we're going to see ADCC. And then at the bottom, we're going to see helminth with IgE. So IgE attracting eosinophils, which can be used to destroy helminths. Excuse me. Plasmodium is fought using where would antibodies come into play? Isn't CMI going to make? So CMI, so when I say CMI, Jacob, with the Plasmodium, CMI being if Plasmodium is intracellular, which a lot of Plasmodium is, because Plasmodium is going to be one of those that cause malaria. So it can be, once it invades certain cells, we can release certain antigens and then experience that and go through CD8 cell activation. Also, we can still get CMI with antibodies. So that's the whole idea behind ADCC, is that we are getting cell-mediated immunity with toxicity, but it's being directed by antibodies attaching onto those antigens or to those microbes. So I know it's confusing because there's a little crossover here. Like at this point, we've never mixed that idea between CMI and antibodies, but that's what the idea of ADCC is. Maybe that answers your question. So some immunodeficiencies. So antibodies bind. Yes. Yeah. So if you think about it, that's how antibodies work. That's what we were saying before in those the different strategies that antibodies do. They're when they cause cytotoxicity, that's going to be them triggering a cell-mediated response, which, like we said, can either be an NK cell, a CD8 cell. There's a lot of cells that can actually induce cell death or induce cytotoxicity, for that matter. So immunodeficiency diseases, these are very important to note. She's going to go over a primary and secondary example. There's also a table in there that kind of explains how these different immunodeficiencies work and the results of them. So that's pretty important to note, and it's a good way to test yourself on your knowledge up to this point. So we have two different types. We have congenital and acquired. So acquired being HIV infections. We talked about this. This is acquired because it's not something inherent in your germline. What it is just you get the HIV infection, and then that leads to a decrease in CD4 cell count. And as we know, if we have decreased CD4, that's decreased helper T cells, So that decreases our ability to make antibodies through isotype switching. So then we have a much, much weaker response. And then a congenital, so a primary. There's multiple examples of these. This is the table I was talking about here at the bottom. This table is great for testing your knowledge on how things work at this point. It's also very important to know for the upcoming immunodeficiencies. So if we have B cell defects, and this is kind of similar to what we were saying with HIV, if we have B cell defects specifically, that means we're not going to be able to deal with exogenous antigens, because that's what our B cells deal with. They deal with those exogenous antigens. So if we have that defect, we can't deal with bacteria because that's exogenous. We also can't deal with protozoans. If we have a T and B cell deficiency, so we're losing both, then we have a problem with not only exogenous, but endogenous. So that's super important to know. You can see T and B cell deficiencies or defects cause problems with our entire immunity. We have no way to respond. So we can't make antibodies. We also don't have that cell-mediated response. The only thing we have is our innate immunity, which is not targeted at all. And then we have another few there, phagocytic disorder. So we see here that if we have a problem with phagocytes, then we can't deal with bacteria, which that should make sense. We can't go through APCs and MHC class 2 presentation because we can't actually get that antigen, engulf it, break it down, and present the antigen. And then we have a problem with fungal. So we saw that one of our two biggest players with fungal infections being macrophage and neutrophils. So some of the examples, make sure you know these. Definitely make sure you know these examples of immunodeficiency, these congenital ones, and make sure you know what they affect. So SCID, so severe combined immunodeficiency, this is a deficiency in both B and T cell. So if you look back at the chart, think about that. Think about what SCID can do and what kind of defects you're going to have in immune responses. It's important to note also that 50% are X-linked and 50% are autosomal. And then DiGeorge syndrome, we had a question on this recently that I created for you all before you kind of saw this information, before we went through it. So DiGeorge syndrome is a underdevelopment or complete lack of a thymus. So you should be able to dig back into your brain and remember thymus is where education happens for T cells. So if we're losing that T cell education, we're going to have a strong difficulty with viral infections because that's our direct way to deal with viruses. And then here, Bruton's, this is something we also had a mention. I had a mention in a question before, and it was way before y'all had seen it. But it is important to know Bruton's is an issue with bone marrow. So you're going to have lack of B cell maturation. If you have lack of B cell maturation, like we said before, you're going to have an issue with extracellular bacteria infections. So it's important to be able to link what's occurring in these, if they're X-linked, if they're autosomal, where the deficiency is in the body physiologically, and where the deficiency is in our immune response. So make sure you can note that in all of these different deficiencies. And then we have an innate immune deficiency. So think about that. This is going to be our phagocytic one that we mentioned in the table. So a phagocytic deficiency, we're going to have a problem with bacterial infections. These are extracellular infections. All right, and then there's our summary, and we'll go on to our next lecture. Lecture 7. Yes, this one. So I do have it broken up into 2 different lectures. I thought that was going to be a little easier, and I know that's how they had it on Canvas, even though they did it all in one. There's no slides missing. They had 50 slides, and we have 22 and 28, so it's still all the same. So here we're going to talk about immune modulation, and we're going to go through a few different strategies that we have, that scientists and physicians have now, that are still in research phase, and then some that are actually FDA approved. Here with the lecture objectives, it's important to note she didn't directly mention certain things when she went through this, so she didn't tell you what molecular therapy is and examples of molecular therapy. What we know molecular therapy is going to be the monoclonal antibodies, so the mAbs. And we'll get into that a little bit more, but it's important to note because she didn't directly outright say that this is her example of molecular therapy. All right, so immune modulation, this is going to be altering or regulating the immune response, and we have specific ways that we do that, and we have specific reasons. So autoimmune diseases, if you think about that, we have an increase in the immune response, so we actually want to decrease the immune response. Same thing with transplantation. If we give you an organ even though we've looked at those HLA markers, there does not necessarily mean that we won't have a response, a graft versus host response. So to prevent that, we use immune modulation to decrease the immune response. Cancers and infectious diseases, both of these, we're going to want to increase the immune response so we can have a better response and deal with the problem, whether it be cancer or certain infectious diseases. So here are some examples. Once again, downregulation is going to occur in autoimmune diseases and transplantation. Downregulation is just going to be an increase in Tregs. Make sure you note the difference, though. With organ transplantation, we're increasing Treg action. With autoimmune, we're creating more Tregs. So the medication that we're using in autoimmune diseases is to create more Tregs. And then with transplantation, it's actually to increase the action. So make the Tregs a little bit more active and responsive. And then with upregulation, with cancers, we're going to see an increase in the immune response either by increasing cytokines, giving the patient antibodies, increasing the function of APCs, or increasing the function of cytotoxic T cells. And we'll go over some examples, mainly the antibodies and the cytotoxic T cells. With infectious disease, this is going to be an increase in the adaptive system with the use of vaccines. And that's going to be the entire second half of this lecture. So immunotherapy, some ways that we can go through immunotherapy to either enhance or suppress the immune response, so how we actually go through immune modulation. We have checkpoint inhibitors, and we're going to go through each of these individually just like Dr. Prost did. But checkpoint inhibitors are going to slow down the immune response by inhibiting those checkpoints that allow the immune system to replicate. So it works just the same as a checkpoint in mitosis. So if we inhibit those checkpoints, then it stops the mitotic division. So it stops us creating more immune cells. And then we have two different types of T cell therapy we're going to get into. And then MABs, monoclonal antibodies. These are very, very big right now. They're huge. Any commercial that you see on medication, look for when it shows up. It'll have the brand name, and then it'll have the generic name underneath in parentheses. If you see MAB at the end of that generic name, it's a monoclonal antibody. And we see these a lot, not only in cancer treatment, but we see these a lot in other autoimmune treatments, so eczema and things like that. And anything that we can benefit by having either a stronger immune response or a decreased immune response, we can use a MAB for. And then vaccinations and then modulators that can enhance response to cancer. So our checkpoint inhibitors, rituximab is a perfect example as a MAB, and that's for lymphomas and leukemias. You'll learn that in pathology and pharm. Sorry, that took me a while. All right, so checkpoint inhibitors. So an immune checkpoint, like I was saying, it's the same thing as a mitotic checkpoint. So the idea being is if we can get past this level and the checkpoint goes through, then we can allow the cell to enter into cell division and create more cells. That's something we see, we didn't really talk about, but we see with the B7 and CD28 interaction. If you remember, the B7 CD28 interaction increased IL-2 and IL-2R, and IL-2 interleukin 2 was used to go with to induce the T cell into cell proliferation. So it's actually forcing it into mitosis to create more cells. What we can do is use a checkpoint inhibitor to actually express CTLA-4. CTLA-4 will bind with the CD28, and it will actually stop that. So remember, CTLA-4 was how we shut down our immune response. So that's going to be one of our checkpoint inhibitors, is using a medication that can increase CTLA-4. We can also use ones that use PD-1L. So PD-1L is actually something that we're seeing a lot in as a target for cancer treatments right now, and they use MABs to target this. And she has a diagram on it a little further down. So PD-1L can be used also as a checkpoint, and it can prevent the cell from going into any sort of response. So here we can see that if we have PD-L1, that's going to prevent our immune response. And there are a lot of tumors, a lot of cancers that are very known for having this PD-L1 receptor on their surface. And if it has that, it's going to inhibit that T cell response. So what a lot of scientists are doing and what a lot of researchers and pharmacists are doing is creating antibodies, monoclonal antibodies, that attach onto that PD-L1 and prevent the interaction between PD-1 and PD-L1. By preventing it, then the T cell can actually interact directly with that tumor cell and induce apoptosis. So this is something that you can see a lot of, and this is something that we are going to be seeing a lot of as future clinicians with treatments that are coming out. So our T cell transfers, we have two different types. We have TIL, so tumor infiltrating lymphocytes, and we have CAR T. There are some staunch differences between them, and we'll go through it. It's actually very, very easy when you look at the specific differences. So TIL, what it is we have cells, we find cells that have infiltrated a tumor, and we take those, and they might be there. There's T cells that might be there but not doing anything. They're not actually activating and attacking these tumor cells. They're just there. So what we do is we take them out and we force them into activation states. So we actually give them the co-stimulatory responses that they need to turn on these T cells. And then we grow more of them. We grow more of these activated T cells, and then we give them back to the patient. Now, this one is still experimental. The reason is because it's not as targeted. So it can have the problems with bystander effect. So by activating these T cells and just giving the patient these T cells that are activated, we could be introducing other side effects. We could be introducing T cells that might just kill anything. So they're not as targeted or as specific. Unlike with CAR T. So with CAR T, we actually get T cells that have infiltrated the tumor. We take them out and we add a gene into them. And that gene is to actually produce this CAR, this chimeric antigen receptor. So the CAR is specific for each person's cancer. So when they do a biopsy, they're going to do flow cytometry to look at the receptors on the outside of the cell. And when they look at those receptors, they're going to see that certain receptors are present on your cancer cells. And they're going to create this chimeric antigen receptor that can bind to that receptor on the cancer cell. And they're going to implant that receptor onto these T cells. Then they're going to grow more and more of these T cells. So now we have a whole army of these T cells that have a specific receptor that binds almost exclusively the cancer. It is almost exclusively because with this you can also still get some autoimmune issues, but it is a lot more targeted than TIL. Now the difference between them, as you can see with TIL, what we're doing is we're just taking it out and activating the T cell. With CAR T, we're taking it out and putting a specific receptor that can target the cancer onto it. So that's the main difference. With TIL, we're just activating. CAR T, we're actually changing the T cell to more accurately target the cancer. And you're going to see here's a nice little diagram that she put in of what happens in CAR T. Now, CAR T is pretty important, mainly because it's one of the big namesakes to Moffitt, which a lot of our professors work directly with Moffitt in their research. So this is kind of a big thing right now, and this is a new way that we are using to fight cancer. The problem with it is it's still extremely, extremely expensive, as well as we do see a lot of responses with autoimmune. And it's kind of tough with both of these, with TIL and CAR T, to go through the normal autoimmune treatments, because you're actually trying to elicit immune response. So if you give this person, if you have someone going through CAR T and you give them a whole bunch of steroids to reduce their immune response, well, what's the whole point? Because now you're reducing their immune response when you want a high immune response. So I have a question in the chat. So if the checkpoint inhibitor stops the response from being too strong, does it selectively target any checkpoint that causes mitosis, or is it specific to? So for T cells, the checkpoint that's actually inducing mitosis will be the IL-2 and IL-2R interaction. So that's going to force it into mitosis. So instead of allowing that to occur, it's going to it's going to force expression of CTLA-4. So that way we stop that IL-2 and IL-2R interaction, which is a checkpoint. So once that checkpoint is marked, once we have IL-2 and IL-2R interaction, that is a checkpoint to allow the T cell to enter into mitosis. So by stopping that with CTLA-4, we're actually stopping that checkpoint from occurring and stopping that progression into mitosis. Hopefully that answers your question clearly. If at any point I answer a question and you guys are still confused after I answer it, just restate it, and then that way I can go through and explain it. But is the inhibitor specific to a single checkpoint molecule? Yes. So the inhibitor in this case for CTLA-4, the actual checkpoint molecule is going to be IL-2. So if you remember from our diagrams before, B7 and CD28, is going to increase IL-2R. So if we get rid of IL-2R by not having that B7 CD28 interaction, and instead we have CD28 CTLA4, which stops the increase of IL-2R, then it's directly inhibiting the action of IL-2, which is going to be that checkpoint molecule.
I'm not sure if I was asking the question properly, but like, is one like a checkpoint inhibitor, can it inhibit any molecule that would start mitosis, or is it that specific, like IL-2?
So the example of the one that induces CTLA4 is going to inhibit specifically the action of IL-2. There are other checkpoint inhibitors. She's not gonna get into all of them, but there are other checkpoint inhibitors that can stop other molecules that actually will act as checkpoints. But the ones that we're talking about here are going to be specifically the CTLA4, which is going to inhibit the action of IL-2 by getting rid of the IL-2 receptor. If that makes any sense. So it's not going to be widespread. These will be very specific. So CAR T therapy success, it is FDA approved for some blood cancers, as well as it's being used in, I believe, final stages of clinical trials for lung cancer at Moffitt. Some of the side effects that you do see that cytokine storm, which can be pretty dangerous, causing a lot of autoimmune issues. So they're not perfect, but they are working on that. So then MABs, monoclonal antibodies. So these are antibodies that are synthesized in a lab. What that means, and the whole process that we actually go through, is you find out an antigen or a receptor that an antibody can bind to. And we use different ones. So because we have a set amount of genetic variation, we actually use different organisms. And usually it's a mouse model, but it can be things other than a mouse. And what we do is we find the genetic variation in those other organisms, and we take that FAB region and attach it onto a human FC region of an antibody. So it's two regions separate, and we combine them together. When we do that, we now have one that will not have the FC region will be able to interact with our immune cells, but the FAB region, because of our limited antigenic variation, we have a wider spread gene pool that we can use for the FAB region, so we can attach onto different epitopes or different receptors. So once the tumors are identified, we can create these MABs for that. So a lot of them, some of them work on like breast cancers. Some of them, pertuzumab, I believe is the name. It works on breast cancers. It attaches onto a HER2 receptor, which is on the outside of certain breast cancer cells. So it attaches onto that, and then it can identify it once again for ADCC, or it can work through a process of neutralization where it just stops the cell's activity. Most of these MABs, when we're dealing with cancer treatment, are going to deal with ADCC, and then some of these other ones that we see in immunotherapy with downregulating the immune response are actually going to go through neutralization, and that's something that's pretty important to note as well. So it's used in cancer, but it's also used in more than just cancer. And this is just an example of the different types of antibodies. So we have a polyclonal, which is something that we do, and it's a lot easier to make. So we make polyclonal, as in we'll make one that has specific AB regions, and we'll make different AB regions for what we can make, what our gene pool allows. And then we have the monoclonal, which is making one specific antibody for one specific antigen receptor. And it's a lot more expensive because you have to go through that process of finding a different gene pool for the FAB region, this little blue region, and then combining it to the humanized FC region. But it is showing to actually be very promising and have a lot less side effects than most other treatments. Here's another image of using these monoclonal antibodies. So once again, the FAB region that we have that can either be humanized, it can be through a mouse model, we'll have a different variation that can attach onto these different receptors, and it'll trigger and cause the actual immune system to attack that cell. So this would be an example here of ADCC through a monoclonal antibody. Some targeted therapies, once again, it's the same idea of using MABs for targeted treatment. Here's rituximab right here for B cell, for lymphoma. So it's going to attach onto the CD20 marker that certain types of lymphoma has, and it's going to force lysis of that cell. So it's a much more targeted treatment than widespread chemotherapy, because it's not actually going to attack any of your red blood cells, or it's not going to deal with any of the nail bed or your hair follicles, anything like that. It should just only target these B cells that have these CD20 marker for rituximab. Then cancer treatment vaccines. This is kind of a new thing that, well, they've been studying it for years, decades actually, but it's something that we're seeing kind of start a little bit more for cancer treatment vaccines. What it is we get, we can either get the patient's tumor with certain antigen markers, or we can use dendritic cells, so APCs with certain antigen markers, or we use tumor-associated antigens, so ones that we know are already associated with these. And we can try to create a mixture of vaccine that we can actually administer to a patient so that it will elicit a better immune response. Because remember, the biggest thing with cancer is it's actually evading the immune system. So if we can find a way to target the immune system with a vaccine, then we can induce that immune response to specific markers on these cell surfaces. I know there are a lot of researchers that are actually going through this route. Some at USF, some not at USF. There was one who was at USF who was doing it on pediatric lymphoma and trying to find a vaccine for that using certain markers on the lymphoma cells. So this is something we're going to see a lot of in the future as well. So these modulators, they can enhance the immune response against cancer. These modulators are used a little less frequently because we have these other treatments that are more specific. Modulators, they can affect specific parts of the immune system, but it gives an immune response that's a lot more general. It's not targeted. So we have the cytokine therapies. By increasing interferons and IL-2, we can increase the immune response. But then with that, we can get autoimmune issues, and we can also get cytokine storms and things like that. Because when you are using cytokines as a treatment, you have to use a very, very high dose of cytokines to actually elicit a response. And then we can see some cytokines that we use to actually alleviate symptoms. So immune regulation with autoimmunity. So this is decreasing the immune response. Once again, we're going to be using CTLA-4. Here's another option, or another time that she kind of threw some CDs at you and didn't explain what they are. So CTLA-4 does bind B7. We know that. And it also can bind CD28. Here, CD80 is B7-1 and CD86 is B7-2. You don't need to know that CD80 is B7-1 or that CD86 is B7-2, but make sure you have that in the back of your mind, that B7 does have an alternate name, and that alternate name being CD80 or CD86. She just kind of threw those at you and didn't really explain that. So it's important to know that interaction. And then we also already know that CTLA-4 is found on Tregs. And Tregs are used heavily for autoimmunity to decrease that immune response, as we said before. So there's our summary. And then we will sneak into the vaccine lecture. We'll try to power through this one so then we can get into the questions. And then once we get through the questions, I'll show you all the image of the study guide that I have, the poorly written study guide that I have. And you can see that. Is chronic inflammation related to type 3 hypersensitivity in any way? Yes. So we can get chronic inflammation, to answer Jacob's question in the chat. We can get chronic inflammation with type 3 hypersensitivity with those antibody-antigen complexes, because those antibody-antigen complexes, wherever they stick, will then recruit other inflammatory cells, so other macrophage and other neutrophils, which will then cause inflammation. All right, so vaccines for infectious diseases. With Dr. Anderson, make sure you note all of his objectives. His objectives are extremely on point. He uses these to make his questions. When I'm making my practice questions, whether it be for these review sessions or the Friday session, this is what he stresses. He stresses his objectives. And you're going to have him for a good amount of the next block. So make sure you go through his objectives. He's telling you right here the generalized topics that he will be asking questions on. So make sure you note those. So here, most of this is just for reference. Really what he wants you to know here is that Pasteur is the one who did everything for starting vaccinations, and it really all started with anthrax. And after you go through this, make sure you kind of understand what type of vaccine he did, what type of vaccine he created for anthrax. And you can kind of get that just by reading this once you understand the different types of vaccines. So the whole purpose of vaccination is to attempt to provide or induce or boost a protective immune response. We do that with one person to prevent disease in one person, but we do that in a community or as an entire population to then provide what's called herd immunity. So herd immunity, and he kind of explained it, herd immunity is when we get about 80 to 85% of people in a population to be completely immune, to already be vaccinated and be completely immune of that disease. And the purpose of that is so that way the other 20 to 15% of people don't have to worry about it. And this 20 to 15% of people are not people that choose to not get vaccinations. They're people that can't get vaccinations, whether it's someone who's going through chemotherapy and doesn't have an immune response to actually create the antibodies or the memory, or it's a child. So a newborn infant hasn't developed enough of an immune system to actually be able to fight off these viruses. And if we expose them to some of these vaccines, then at that early age, it can actually have adverse effects. So things just like, getting them sick or weakening their immune system more and exposing them to and making them more vulnerable. So the idea behind herd immunity is to have the majority of the population be completely immune to these viruses, which then protects those people that can't get vaccinated. So it's important to know that that's the idea behind herd immunity. And a lot of your professors are going to really stress that, because that's what a lot of the anti-vaxxers the anti-vax community is not getting, is that the reason we vaccinate is not just for you as an individual, but it's for the people that can't get vaccinated. And we do that through herd immunity. All right, and make sure to note his questions. He's never going to use this direct question. Well, I can't say never, but for our year, he didn't use any of these direct questions for his tests. But these are good checkpoints to see where your knowledge is at. And he'll have more of them when he actually does the bacteriology lectures. So passive immunity, we've gone through this a lot. Passive immunity is temporary. It can be done post-exposure. And what it really is just giving a person immune globulins. So you're just already actively giving them immune globulins, and they're not creating their own antibodies. You're just giving them the antibodies to protect them. So it doesn't give any long-term immunity, but it is used for immunosuppressed patients, people that can't create their own antibodies. And these are some examples. Make sure to know these examples. So here with the second example, passive transfer of IgG from mother to child. That one's pretty important because the IgG can cross the placenta. It's the only one. So make sure to note here, I didn't write it down, but it's IgG. And then in breastfeeding, it's IgA. So when we go through, when we actually go through breastfeeding, because it's a mucosal surface, the mother is transferring IgA to the infant for another form of protection. And then active immunization. So this is delayed, but it is more permanent because you are creating your own antibodies. It's important to note this is what vaccines do. So vaccines are forcing you to create your own antibodies, so that way you can have that permanent immunity to whatever we're trying to protect you from. These vaccine characteristics, these are extremely important as well. Effectiveness, availability, and stability. Dr. Anderson went through this and he explained the importance of all of these. So stability, so that way you don't have to worry about refrigerating it. You can take this vaccination anywhere you might need in climates that are very, very harsh. So it needs to be stable enough that it can travel and deal with that. It also needs to be available and cheap. This is kind of the big thing that we see a lot of vaccines go through in the final stages of clinical trials. And then the initial stages of clinical trials are going to be the effectiveness. So how well does it work? How much immunity does it actually give the person? Our types of vaccines. So we have multiple different types. We're going to go through them. This is going to be the biggest bulk of the lecture here. So an inactivated or killed vaccine. This is relatively safe. What that means is we can give it to the host without too much worry or concern. Make sure you know with all of these vaccines what type of antigen is being used, what type of immune response is being activated, and then really, really know these examples. So make sure you know for inactivated, it's the Salk polio. You have two different types of polio. So this one's the Salk polio, the inactivated flu, hepatitis A, and cholera. These are your examples that they want you to know for sure. But really make sure you know that list of examples, the type of antigen being used. If you know the type of antigen being used, you should be able to assess and immediately assume what type of response we're getting. So if it's exogenous, then we're getting antibodies being produced, because exogenous is going to create MHC class 2 response, which is then going to interact with CD4 and B cells interacting. You get class switching, which gives you a better, stronger antibody. So the live vaccinations. They are weakened preparations. So what we do is what scientists do is they get the live strain and they weaken it. So they take out certain virulence factors. So that way it should not infect you. But there is always a potential for reversion. This can happen. This happens a lot more frequently with the oral Sabin polio vaccine. A lot of pediatricians aren't giving that anymore because of that reversion. I think they stopped giving it in the early 2000s is when they had some of their last cases of it. Here the examples being varicella zoster, so chickenpox, measles, mumps, rubella, and the oral Sabin vaccine. This one gives you an endogenous and exogenous response. So it's giving you both. So you're getting exogenous, which is creating antibodies, and endogenous, which is creating your cellular response. So it's going to give you their strongest response as a vaccine because you're getting both of those. So that's very, very important to note. Our toxoid, he talked about, Dr. Anderson talked about this a decent amount. Toxoid, so what they do is they get a toxin from a specific bacteria. So make note that this is bacterial toxins. They get that toxin and they treat it with formalin. And by doing that, they create what's called a toxoid. So the toxoid still produces an immune response, but it actually has no effects on your cells. So they've removed that effect on the cell, but we still retain that immune response. The examples of this being the tetanus and diphtheria toxin. We're going to talk about the diphtheria one later on towards the bottom of the lecture, but the tetanus and diphtheria toxin, make note once again, exogenous, so it's giving you antibody production, which makes sense because we want antibodies to attach onto these toxins and neutralize them. So our subunit, we actually get DNA from the virus or the bacteria, depending on what we're actually trying to immunize for. In this case, we're using pertussis, so Bordetella pertussis, so whooping cough. We get a piece of its DNA, and we actually inject you with that. With that, that's going to give us, because it is exogenous, it was not made within a cell, within our own cells, we get the exogenous response, which creates antibodies. The recombinant protein, so this one is similar to the subunit, but instead of it being DNA, we take a protein. We take that protein from that virus or that bacteria, and then we reverse engineer DNA from that. So just like in the subunit, we're using DNA, but here it starts with a protein. So then we make that DNA, we inject that DNA, and that can give immunity. The examples here, Hep B, as well as recombinant influenza. Another example that was brought up in the lecture that someone asked in the chat was Gardasil. So Gardasil, they do the same thing. They take the proteins from each strain of HPV, and they create a recombinant DNA. So it's important to note here with recombinant protein, and the reason that we have the recombinant influenza one here is these recombinant ones allow for vaccination from multiple different strains of the same virus. So influenza, we have multiple different strains. We have the H1N1, we have type A, we have type B. With this recombinant influenza vaccine, we can actually incorporate all of these different strains because we're incorporating different proteins from each one and reverse engineering that DNA and administering that DNA to you. So now you can build a response to all those different viral strains that are in that vaccine. And once again, this one's exogenous, and it's going to create an antibody response. As you're seeing, most of them are exogenous and do create an antibody response. Our conjugated polysaccharide vaccine. So we get a polysaccharide from the bacteria, and what that actually is going to do is that's going to elicit a response, but because it's a polysaccharide, that will elicit a T cell independent response, which only allows us to make IgM. So instead, what they do is they attach a protein conjugate onto that. So they attach the protein conjugate onto the polysaccharide. So now we can elicit not just a B cell response, but because that protein, we can elicit a T cell dependent response. So remember, if we have that protein and the peptides, we can elicit that T cell dependent response, which will allow us to go through class switching. And class switching is what's going to allow us to go from that initial IgM to create the IgG or IgA, whichever one we might need. Examples of this, Haemophilus influenza B, which is found in your pediatric regimen, pneumococcal and meningococcal. So pneumococcal being bacterial pneumonia, and then meningococcal is bacterial meningitis. And then this one, we're going to have exogenous antigen as well that creates antibodies. So if you have a live attenuated vaccine of bacteria. So that one actually wouldn't, so if you're just administering a live bacteria, that really wouldn't create a vaccination. That would just be infecting the person with the bacteria, and it would create an exogenous antigen response. But that's just our normal bacterial response, and we're constantly being exposed to that, which isn't. It's very hard to build long-term immunity to some of these bacteria, and I know that seems confusing when that can occur with a virus and not as much with a bacteria, but you'll get into that with Dr. Anderson when he explains kind of the antigenic variations and some of the defenses that we've already talked about that bacteria have for that long-term immunity. So some experimental vaccines that we see. I have some of these written down on my study sheet, but not all of them. Mainly, I don't have the RNA because this is something that's very new. Dr. Tang has been working on this. He's going to lecture in the virology section. And Dr. Anderson stressed that RNA is one of the things that they're using for COVID. And they essentially they inject direct RNA, so that way then the RNA is in you and you can produce the protein inside, which will give you an endogenous response. And then we have DNA. None of these are actually directly approved for human use at the moment. They are working on them. The recombinant vectored vaccine is using a retrovirus, so adenovirus, lenti, and vaccinia virus. They're using those so that way we can create that response to these to certain other viruses that are a little harder to do. So we use retroviruses in that one. So adjuvants, we've talked about this before. We've already talked about this, and we've already introduced the topic. So adjuvant is just a non-specific immune enhancer. So what that means is it's something we're adding to these vaccines, and we usually add them to any non-live vaccine. So the toxoid, the polysaccharide conjugate, the recombinant DNA, all of those are going to require adjuvants. And the reason we use that is so we can elicit a stronger immune response. We can elicit that stronger inflammatory response. So we can attract more neutrophils, more macrophage, and then even more dendritic cells. So that way we have more APCs to interact with that antigen that we're administering. So we can get a stronger response and build stronger immunity. Make sure you know these different types of adjuvants. That's going to become something pretty important. Or inorganic salts, and then oils, bacterial products, or aluminum hydroxide. These are things that are very, very important to remember as adjuvants because we will talk about these in different ways, and we will have examples of them. So to get to our examples here are 3 examples at the end of the lecture. So tuberculosis. We've talked about this a little bit already. TB, when we talked about the type 4 hypersensitivity. So tuberculosis is caused by Mycobacterium tuberculosis. It's a very slow-growing disease, and you can see in this X-ray down here in the left lung, you see a lot of fibrosis inside the lung, and that's a pretty indicative sign of tuberculosis. So tuberculosis, so some of the immune responses that can determine the course of the disease, Th2, we already know that Th2 increases an inflammatory response rather than a cell-mediated response. So because it's introducing an inflammatory response, that's going to increase the progressiveness of the disease. It's going to make the disease last a lot longer. And then the Th1 is going to introduce a cell-mediated response. So it's actually going to help to protect you from any sort of tuberculosis infection. And then here, there is a vaccine for it. As Dr. Anderson was expressing, this vaccine is not used in the United States, and he talks about that, and we'll kind of review why it's not used in the United States. This BCG vaccine. So it's cultured actually from a different strain of Mycobacterium. It's cultured from Microbacterium bovis. And what someone did was they actually went through and bred this strain of Mycobacterium bovis over 13 years to actually make it avirulent. So it can still elicit immune response, but it doesn't have any efficacy to its response. It's actually not going to cause any sort of damage to your cellular tissue. Now, the problem with this, and we're going to get into it, is it doesn't elicit the best response that we need for a vaccination. So the cost doesn't outweigh, or the benefit doesn't outweigh the cost in this scenario. So once again, what it does is it provides immunity for CD4 cells. So it creates an antibody immunity. So the limits to it. So there were initial deaths with it because you are introducing a bacterial strain into the body, and that can cause those cytokine storms and the different hypersensitivities or autoimmune issues that you see. The 2 bottom bullet points are probably the biggest one that has varied efficacy. So it didn't provide the immunity that we actually expected, or that scientists expected, which should make sense from your point of view at this moment, because it is not tuberculosis. It's actually it's a mycobacterium, but it's bovis, so it's a different strain. So it will protect you in generalities to certain epitopes that you see on all mycobacterium, but not on things that we see on tuberculosis specifically. And then the other big thing, and this is why we don't use it in the US, is because it compromises the TB test. So it compromises that type 4 hypersensitivity test because you've already been exposed to mycobacterium. You now have antibodies to that. So you can get that antibody response, that cell-mediated response that you get in type 4, because those antibodies are going to attach onto it, and we're going to see a lot of killing and a lot of death. And remember, when we see that ADCC, we also see an influx of inflammation. So you should remember the picture of type 4 hypersensitivity where the person had the TB test, and you saw their arm was like very red and inflamed, and you saw a little bit of cell death going on. That's something that you can see with someone if they've received the BCG vaccine, if you do a TB test. So instead of doing that, what they usually do is they do a chest x-ray for anyone that might have already had tuberculosis or might have had the vaccine or been exposed to tuberculosis. So that way they can avoid that hypersensitivity and they can see if they have active TB or not. So our second example, diphtheria. So diphtheria being a bacteria, we'll talk more. Some of y'all kind of already have an idea of it, but we'll talk more about gram-positive and bacilli, the different shapes and the different gram-staining reactions. But diphtheria creates an exotoxin, so a toxin outside of itself. And that exotoxin actually inhibits protein synthesis. So we use antibodies to neutralize that, and this is in that toxoid vaccine, that diphtheria vaccination. Here's just an example, an image of kind of what it looks like when you have that buildup of that toxin creating this film. So we use a toxin. We use that toxoid vaccine. That the way we create it, same way we would create any toxoid. We get the toxin, we treat it with formalin, so that it turns that toxin into a toxoid, and then it's then added with an adjuvant. That adjuvant here being aluminum hydroxide. That adjuvant can then elicit that immune response. That way we can create the antibodies to neutralize that toxin. And our third example, our last example, is Bordetella pertussis, so whooping cough. Here, this one, it's gram-negative, aerobic, and it's non-motile. There's just some different facts that you'll have to get into a little bit more. And then it causes whooping cough, which is necrotizing inflammation in the lower respiratory system. As Dr. Anderson did extend to you all three of these are very, very infectious. These are very, very dangerous pathologies. With that, we have virulence factors that are linked to Bordetella pertussis. We have the toxin, which is also another exotoxin, but this one inhibits cell signaling. And then we have protactin, hemagglutinin, and fimbriae. These are all for attachment. These are all three different attachment proteins that actually allow this toxin to interact with cell signaling. So these are all things that we can use and target in our vaccination. So Shreya, so does non-active TB show up on chest x-ray? Is that the fibrosis? So non-active TB should not show up on an x-ray. The fibrosis is going to be something that's currently active in TB. So if they do see any sort of fibrosis, even if you have non-active TB, and when they do the chest x-ray, they'll put you on an antibiotic regimen. It's a whole combination of like 4 different antibiotics, and you go through that whole cycle for a while before you can be allowed to do whatever you were going to do, whether it's work in a hospital or in med school, do certain rotations, because you have to be free of TB before you can go into a hospital as a student or in shadowing or as an employee. So they'll do the chest x-ray. If they see anything that's questionable, then they'll put you on the antibiotic treatment. But non-active TB should not show up on an x-ray now. So the pertussis vaccine, we had 2 different types. We had the old vaccine, and then now we have a toxoid vaccine. This is actually added on to our diphtheria and tetanus. It's called the DPT, and it's in the childhood the pediatric regimen. So it does elicit a very strong Th1 response, which is going to increase IL-2 and interferon gamma. And we already know what IL-2 and interferon gamma do by enhancing our immune response. And then here, these are his study questions at the end. Usually his study questions are more actual direct questions with multiple choice answers. Make sure you go through those. Make sure you go through this. So these actually have things that link directly right back up to your objectives, but it has other things as well. So like what we explained with BCG, why it's not used in the US, how it elicits immunity, what is herd immunity, all these things. Make sure you kind of go through these Make sure you know the answers to these before going into your test. Like I said, I can't stress enough how important it is to make these study sheets and note sheets now and not wait until after you've already done exams in your block. You want to have as many of these study sheets done before your anatomy exam next Thursday. All right, so now we'll go into our poll everywhere. I am not able to see the chat, so if you are asking questions in the chat, sorry, I can't actually see it right now, but everyone should know how to enter into the poll everywhere. Go ahead, it is active. It is active. So you can start and we'll go through them. There's only about 6 questions. Once we go through all of them, then I'll show you my study sheet. I'll answer any other questions, and then that'll be it for the night. Oh, I really like the top one. Yeah, I really like the top one a lot. No, I'm not muted. That's fine. They're doing the question. And even before I lock it, they're not doing well. I'm gonna lock it before all y'all can change your answers now that I just said that. Yep. All right, so 79% of you, 77% of y'all got it wrong, even though 90% of y'all technically got it wrong, and then you heard me say that you weren't doing well and change your answers. So memory T cells require CD28 and B7 interaction in order to be activated. So the big thing here that I should point out to you is memory T cells. Memory T cells only need initial presentation. They don't need that co-stimulatory response. Same thing with memory B cells. They've already gone through that initial exposure to antigen, so they don't need that co-stimulatory response, because they know once they see that antigen and that antigen is presented to them, they know we've already dealt with this. We know exactly what to do. They don't need to be told, hey, release IL-2 and IL-2R, so that way you can stimulate and you can create more of yourselves. So make sure you note that. This is a little tricky one that I kind of threw in there to you all. But whenever you see memory B cells or T cells, they only need one activation. They don't need co-stimulatory activation. They just need antigen presentation. They don't need that second stimulatory activation to activate, because they've already gone through it. They've already experienced it. Also, before I go on, I would like to note, as always with every question I make, all of these have been vetted through Dr. Anderson and Dr. Pross. So these are all direct topics that they want you to know, and these are all things that are important for the test. Okay, let's go on to the next one. Remember, I'll answer any questions that y'all might have after, because I can't see the chat. Should be active. Yeah. All right, I'm gonna give you all probably another 10 seconds. I gave you all a long time on that last one, but because I wasn't paying attention. This is important. All right, get ready to lock this one up. So what leg of the immune system is being activated by the use of vaccines? So remember, vaccines, the entire purpose is to give you an antigen in any different form so that you can create your own antibodies. So if you're creating your own antibodies, then yes, it is the adaptive immunity. Remember, passive immunity is if we just give you the antibodies. We're just giving them to you. Make sure when you go back through these questions when you're studying, as well as all the questions for your other classes, don't just look for the answer. In this one, as an example, either explain it in your study group or write it down if you're studying by yourself. Why is innate immunity not an answer? Why is passive immunity not an answer? Things like that. So don't just look for just the answer. When you're reviewing these, go through on what the wrong answers were and why. It's going to really help you learn the material better.
Hey, Daniel, could you share your screen again?
Are y'all not seeing my screen?
We can't see what the right answers are when you finish when you're explaining.
Huh? You can't see anything, or you can't see the right answers?
like after when you close the poll and show us what the right answer is, like we can't see that.
Yeah.
We can see your face.
Oh, well, that's nice. I mean, that's something, right? Y'all can see that. All right, how about now? Can you all see everything?
Much better.
Now? There you
Yeah,
go.
now we can see. Thank you.
Perfect. No problem. Sorry, I didn't know. I didn't know you guys were just looking at my face the entire time. Doo-doo-doo.
Now we can see both.
Perfect. Exactly what you wanted to see. I'm gonna give you all a little bit more time on this one. All right, we'll give you about 5 more seconds before I lock this one. All right, so I am locked. So the question being, humoral and cell-mediated immunity both contribute to suppressing tumor growth, but humoral is the most important component. We know that is false because when it comes to tumors, the most important is going to be your CD8 cells and your NK cells, which are all cell-mediated. Remember, humoral is antibodies. So antibody treatment and humoral treatment does work well when we're giving it to you as a treatment, but the actual suppression of tumor growth, what stops you from developing into cancer in your normal everyday life, is actually going to be that cell-mediated, the CD8 cells and the NK cells with the MHC class 1. All right, here is your next question. So this one, I'm essentially just asking what type is MMR, what type is Hep B, and what type is diphtheria. All right, I'm gonna give you a little bit more time, probably about 8 more seconds on this one. And we got some people here that are very weak to group thought. All right, so what are the different types of vaccines for MMR, Hep B, and diphtheria? The easy ones should be MMR and diphtheria. We've talked about these, MMR being live, diphtheria being a toxoid. The question is, what is Hep B? So Hep B being a recombinant protein. Make sure, I can't stress it enough, make sure you go through those examples. Those examples are so crucial when it comes time for the test. Not just the examples, but how the vaccine works. All right, here's your next question. Speaking of how a vaccine works. I know these are challenging considering we just went over these and y'all probably haven't studied which vaccine is used for what, but it is something you need to remember, and I would suggest starting to make those study sheets as soon as possible. I'll give you about 6 more seconds. All right, let's lock this one up. So one of your patients is headed to Haiti for a mission trip and asked you about the cholera vaccine. What do you remember about the cholera vaccine from your MSP3 immunology block? So when we go through it, we should remember that cholera is inactivated. So it's an inactivated vaccine. If it's inactivated, that means it's going to produce antibodies. So antibodies, that's actually what almost all of them do. The only one that we have that you have to study for is going to be the live, which produces both humoral and cellular. But that is not going to be, cholera is not an example of a live vaccine. Here is your next one. How many people look through their notes for these? You should be trying to do these off the top of your heads as a side note. I don't know if, I'm sure some people are, and some people are looking at their notes, but try your best to do it off the top of your head to see how well your memory is working and how much you can remember. If you're not doing as well on these, try to change your strategies up and review the information before class, review it after lecture, and give yourself some time. I'm gonna lock this one up in a few moments. All right, so diphtheria toxin. So diphtheria toxoid vaccine, we already talked about the toxoid vaccines. They are made with adjuvants and toxoids. So here, the toxoid plus the aluminum salt, that's going to be our inorganic salt. It's going to be that adjuvant that we're using here. So that's important to note. All right, and that is our last question for review. Does anyone else have any other questions regarding this information or anything?
There are some questions in the chat.
Oh, thank you for informing me, Jacob. All right. Can you share your screen? We can't see your screen. Okay. I think this is the last one. So for memory T cells to be created in the first place, they needed the secondary. Yes. So for the memory question, I know that got a lot of people. For memory T and B cells, initially, when they are naive, before they go to that form of being a memory T cell or B cell, when they are in that naive state, they need the initial presentation of antigen as well as that co-stimulatory response. After that, they do not need the co-stimulatory response once they've graduated onto a memory T or B cell or a plasma cell. So make sure to note that difference. Bacterial polysaccharides are bad vaccines because they are not peptides, right? Only B cells can bind and produce IgM with B cells. So Jacob, to answer your question, actually, bacterial polysaccharide vaccines, they're not effective, so they will only make IgM. But if we're using a conjugate polysaccharide, that's adding a protein on, which can then induce and allow for the class switching. So that's how we get around that that formality. If by using a polysaccharide only gives us a T cell independent response, if we attach a protein onto that, we can then stimulate a T cell dependent response and get class switching. Okay, it's a specific example, but in this case, he was testing to make sure we. So yes, thank you for answering the question, Ryan. Answering Jacob's question. So it's not that it's I wasn't testing specifically that you knew it was an adjuvant. What it was mainly looking for was, could you differentiate the fact that you needed to know that a toxoid vaccine had a toxoid in it plus an adjuvant. So then we have a whole gambit of adjuvants to look at, whether it be an inorganic salt, aluminum hydroxide, anything. We have a lot of different variations, but just making sure that it was really the toxoid was the thing that I was trying to hit home with that question. So does anyone else have any other questions? Feel free to ask.
I did have a quick question.
Mm-hmm.
It's about the antibody-driven cell.
Mm-hmm. Yeah, ADCC.
Yeah, via IgE and IgG. Yes. I'm kind of confused when each of them are used. Could you clarify that, please?
So they're going to be used anytime we have antibodies that are pre-made for certain antigens. So for instance, if you have a bacteria and that bacteria infects your body, macrophage will come by and phagocytize that bacteria, present its antigens on the MHC class 2. When it presents it on the MHC class 2, then it'll present it to a CD4. That CD4 cell then goes to a B cell, and we get class switching, right? And we get the antibody production. So for let's say in that example, we're creating a specific IgG. So we have that specific IgG. 2 years down the road, that same we come in contact with that same bacteria. Now that we're in contact with that same bacteria, we have a specific response for that bacteria now. We have this free-floating antibody, comes in contact with the bacteria, binds onto it. So it binds with the FAB region. Now that it's been bound, that bacteria is now flagged for cell death. So if we have an NK cell, if we have a CD8 cell, a T cell just kind of roaming around, it can attach onto that FC region and know immediately. We don't have to go through this process of MHC. We don't have to go through this process of class switching. We already have the antibody, and we can target this and kill it right away.
Okay, that's clear. But when do you know to use IgE over IgG, or like that's?
So and that depends on the antigen. So IgE is going to be used for any sort of helminth, so any worm infection, or any sort of allergen, so like a pollen or anything like that.
Okay.
And then IgG is the workhorse. So when you go through the IgEs, IgE is allergy and helminth. IgA is mucosal. And so anything in the mucosa is going to be using IgA. IgM, first one made. IgD is bound on naive B cells. IgG does everything else.
Gotcha.
Literally everything else. It is the workhorse. It's going to do as much as possible.
Okay. Thank you so much.
No problem. Xavier, so will it be a glycoprotein? I'm assuming that's in reference to the explanation to the polysaccharide. Technically, you could consider it a glycoprotein. Thank you for the clarification. Technically, you could consider it a glycoprotein. Be careful with the terminology. Not that it's wrong, but we're going to get into things later on where cells specifically interact with their glycoproteins on their surface, and I don't want you to get that confused with these receptors that we're going to talk about later. But yeah, it's technically still a protein with a sugar with a protein moiety on it. So yes, that technically answers that. Does anyone else have any other questions? Okay, so before I get off, I would like to remind you all that next Wednesday I will not be having a review, that I'm going to be combining, so that way you can have an anatomy review before your anatomy test. I will be combining the 3 lectures, so